Hello everyone. This is Ashraf Hassanpour from Shiraz University of Medical Sciences. Welcome to today's lecture where we'll discuss the anatomy of the cerebellum as well as its functions and clinical relevance. The word cerebellum derives its name from the Latin word for little brain, which is exactly what it looks like. Functionally, the cerebellum is part of the motor system. However, it doesn't trigger any conscious movements, but is responsible for unconscious coordination and fine-tuning of movements. The cerebellum is involved in maintenance of balance and posture. Other functions associated to the cerebellum include coordination of voluntary movements, motor learning, and cognitive functions, for example, in language. The cerebellum is found at the base of the brain. It's separated from the occipital lobe by the tentorium cerebelli, a tough layer of the dura mater, and fills most of the posterior cranial fossa. Cerebellum is anteriorly related to the posterior surface of pons and medulla oblongata, from which it's separated by fourth ventricle of brain. Cerebellum is connected to brainstem by three large bundles of fibers called cerebellar peduncles. Superior peduncle connects cerebellum with midbrain, middle peduncle with pons, and inferior peduncle with medulla oblongata. The inferior and middle cerebellar peduncles are mainly cerebellar afferents, whereas the superior cerebellar peduncle contains mainly cerebellar efferent fibers. This image shows us the superior or tentorial surface of the cerebellum. The external surface of the cerebellum displays numerous folds that enlarge the cerebellar cortex like the gyri of cerebrum. The folds on the cerebellum are too small to be called gyri, and so are referred to as folia, meaning leaves. And it's quite easy to see here that the cerebellum has two hemispheres, the left hemisphere and the right hemisphere. Separating these two hemispheres and running right down the center is a structure we call the vermis. Vermis is so called because it's somewhat like worms in appearance. The cerebellum can be divided according to three different criteria: gross anatomical, phylogenetical, and functional. Let us start off with anatomical division of cerebellum. So have a look at this image over here. Highlighted on the cerebellum is the horizontal fissure. And if we cut the cerebellum along the horizontal fissure and then lay it face up, we end up with a structure that looks like this. So the various parts of cerebellum are shown in this image, where both the superior and inferior surfaces of the cerebellum are drawn in one plane. The upper part of the diagram, above the horizontal fissure, represents the superior surface, and the lower part, below the horizontal fissure, represents the inferior surface. So, as I mentioned, the horizontal fissure, that is the largest and deepest fissure in the cerebellum, separates the superior surface of cerebellum from the inferior surface. Also, we have primary fissure, that is a deep V-shaped fissure and separates the anterior lobe from the middle or posterior lobe on the superior surface of the cerebellum. And finally, you see here the posterolateral fissure that separates the posterior lobe from the folliculonodular lobe on the inferior surface. So as we saw, each hemisphere is divided into three lobes. The anterior lobe lies on the anterior part of the superior surface. It's separated from the posterior lobe by the primary fissure, and vermal portion of anterior lobe consists of lingual, central lobule, and culmen. The posterior lobe is the largest of three lobes situated on both its surfaces. It's limited in front by the primary fissure on the superior surface and by the posterolateral fissure on the inferior surface. Vermal portion of superior surface of posterior lobe consists of declive and folium, while inferior surface consists of tuber, pyramid, and uvula. The folliculonodular lobe is the smallest lobe of the cerebellum. It's located on the inferior surface in front of posterolateral fissure and comprises of nodule of inferior vermis and a pair of folliculus, while are connected to the nodule by peduncles. Also in this view, you can see cerebellar tonsils. They are a characteristic feature of the inferior surface of the posterior lobe, and I'd like to add here an important clinical note associated to the cerebellar tonsils. As you can see in this slide, the tonsils lie close to foramen magnum and are forced down into the vertebral canal when there is a sudden increase in intracranial pressure. This condition, known as tonsillar herniation. Usually, tonsillar herniation is caused by an expanding infratentorial mass such as cerebellar hemorrhage. The cerebellar tonsils force through the foramen magnum, compress the brainstem, and obstruct cerebrospinal fluid flow. The cerebellum can also be divided in three parts based on functional criteria. So, the anterior and posterior lobes are organized into three longitudinal zones: vermis, intermediate, and lateral. So first, we have the vermal zone or vermis, highlighted here in blue, which is concerned with control of muscles of trunk, neck, shoulders, and hips through vestibulospinal and reticulospinal tracts. And just beside this zone, both on the left and right side, we have the paravermal zones or intermediate zones, highlighted in brown, which is concerned with control of muscles of distal parts of limbs like hands and feet via rubrospinal tracts. And lastly, adjacent and on the outermost edges, we have the lateral zones highlighted in purple, which is connected with association areas of the brain and is involved in planning, programming, and coordination of muscular activities of the body. It is done through dento-rubro-thalamocortical tract and descending corticospinal tracts. Phylogenetically, cerebellum is divided into three subdivisions: archicerebellum, paleocerebellum, and neocerebellum. Archicerebellum or vestibular cerebellum is the oldest part of cerebellum and first to appear in aquatic vertebrates. Fishes and lower amphibians possess only this component of the cerebellum. Archicerebellum comprises of folliculonodular lobe and lingula and has mainly vestibular connections. It receives inputs from the vestibular system of inner ear and sends outputs back to the vestibular nuclei. It maintains balance, tone, and posture of trunk muscles and controls ocular reflexes, mainly fixation on a target. Paleocerebellum or spinal cerebellum appears next in in the terrestrial vertebrates with the appearance of limbs. It includes pyramid, uvula, and anterior lobe except lingula. It's concerned with spinal cerebellar connections and responsible for tone, posture, and crude movement of the limb. Neocerebellum or cerebral cerebellum is the most recent part of cerebellum to develop. It develops in primates and associated with the enlargement of telencephalon and cerebral cortex. It's very prominent in the higher mammals. Neocerebellum includes posterior lobe except pyramid and uvula, and it is involved in planning movements and motor learning. It receives inputs from the cerebral cortex and pontine nuclei and sends outputs to the thalamus and red nucleus. This area also regulates coordination of muscle activation and is important in visual guided movements. So, it is concerned with a smooth performance of skilled voluntary movements such as a learning of hit a baseball. Now, let's talk about internal structure of cerebellum. In cross-section, cerebellum is comprised of a gray cerebellar cortex, a medullary core of white matter, and 4 pairs of intrinsic nuclei. So, as I mentioned previously, the cerebellar cortex consists of numerous narrow leaf-like laminae known as the cerebellar folia. The medullary core of white matter has a complex branching system, and together with the tree-like appearance of laminae and folia, it has been given the descriptive term arbor vitae, the tree of life. And deep within the white matter of each hemisphere are 4 masses of cerebellar nuclei. From lateral to medial, they are the dentate, embelliform, globose, and fastigial. Note that the embelliform and globose are collectively referred to as the interposed nuclei. Now, let's focus on the cerebellar cortex and talk about cellular anatomy of cerebellum. Cerebellar cortex composed of 3 distinct layers. Outer molecular layer that contains stellate cells and basket cells. Intermediate layer of Purkinje cells that contains single layer of cell bodies of Purkinje cells. And finally, inner granular layer that made up of cell bodies and dendrites of granule and Golgi cells. Neuroglia are present in all the layers. Fibers entering the cerebellum proceed as massive fibers from various regions of CNS or climbing fibers just from olivary nucleus. Massive fibers form excitatory synapses with the dendrites of granule cells in the granule cell layer. From here, the granule cells send axons to the molecular layer, where they bifurcate into parallel fibers that run longitudinally in the folia. The parallel fibers then synapse on Purkinje cells in the outermost Purkinje cell layer. Climbing fibers project to the Purkinje cell layer and form powerful excitatory connections with the Purkinje cells. The Purkinje cells then project to the deep cerebellar nuclei. All intrinsic neurons except granule cells are inhibitory, and such collection of inhibitory neurons is not found anywhere in the central nervous system except cerebellum. You will learn about mechanism of cerebellar cortical circuit in more detail in physiology section, inshallah. For now, let's move on to look at the cerebellar nuclei that are located close to roof of fourth ventricle on each side of midline. So first, Let's start with the largest of them, the dentate nuclei. Dentate nucleus is the most prominent cerebellar nucleus and the largest in primates including human beings. It's found within the lateral zones of the cerebellum, so it belongs to no cerebellum and receives afferent from it. Its shape is like crumpled purse with hilum directed ventromedially, and its interior is filled with white matter consisting of efferent fibers forming most of the superior cerebellar peduncle. Axons from dentate nuclei project to the contralateral ventral nucleus of the thalamus after decussating in the superior cerebellar peduncle. From here, axons of thalamic nuclei project to the motor cortex. Its primary function is to aid in the regulation of many aspects of voluntary motor activity, and these include inception, planning, and timing of motor activities. Embryoniform and globus nuclei together are known as interposed nuclei. Embryoniform nucleus is oval-shaped and located medial to the dentate nucleus. Globus nucleus is rounded in shape and situated between embryoniform and fastigial nuclei. They belong to paleocerebellum, so receive fibers from paleocerebellum. The embryoniform and globus nuclei have a similar course as the axons from the dentate, but with the addition of another synaptic target. Axons from the interposed nuclei decussate in the superior cerebellar peduncle to synapse on the contralateral ventral nucleus of thalamus and contralateral red nucleus in the midbrain. Axons leaving the red nucleus descend to inferior olivary nucleus in the medulla. The axonal projections from the interposed nuclei function in monitoring and correcting motor activity of upper and lower extremities. The last set of nuclei that we are going to be looking at are the fastigial nuclei. This nucleus is situated in the midline in the vermis and is smaller than dentate nucleus but larger than interposed nuclei. It belongs to archicerebellum, so receiving afferents from it. Axons from the fastigial nucleus pass through the inferior cerebellar peduncle to reach the vestibular nuclei and reticular formation. These in turn result in the stimulation of proximal and trunk musculature to maintain balance. Now that we've covered some structures of cerebellar cortex as well as cerebellar nuclei, let's move on to discuss about white matter of cerebellum, and let's start with afferent fibers. Cerebellum acquires information from cerebral cortex, spinal cord, vestibular apparatus, red nucleus, and tectum of midbrain through afferent fibers. Cerebellum accrues input from cerebral cortex through cortico-pontocerebellar, cortico-olivocerebellar, and cortico-reticulocerebellar pathways. Also, cerebellum receives information from spinal cord through anterior spinocerebellar, posterior spinocerebellar, and conocerebellar tracts, and that from vestibular apparatus either directly or after relaying in the vestibular nuclei. As you see, afferent fibers reach the cerebellum mainly through middle and inferior cerebellar peduncle, and as we discussed, are of two types, massy fibers and climbing fibers. Massy fibers are the main afferent fibers of cerebellum from various parts of CNS like cortex and pons, while climbing fibers arises in the inferior olivary nucleus. let's move on to efferent fibers. Cerebellum provides output to red nucleus, thalamus, vestibular nuclei, and reticular formation through efferent fibers via Purkinje cells. Majority of axons of Purkinje cells synapse with neurons of intracerebellar nuclei, which in turn project to other parts of nervous system. But few Purkinje cells from folliculonodular lobe and vermis directly end in lateral vestibular nuclei. Efferent fibers from dentate, embryoniform, and gilbus nuclei travel through superior cerebellar peduncle, and those from fastigial nucleus through inferior cerebellar peduncle. Now, I'd like to have a quick overview of functions principle of cerebellum. Hopefully this will help you to understand the concepts better. It has already been mentioned, though cerebellum is not concerned with initiation of voluntary movement, it regulates normal motor activities unconsciously. It acts as a playback singer or trainer of a music troupe. Functions of cerebellum is explained in 3 stages. In stage 1, cerebellum receives various kinds of sensory informations either through direct pathway like spinocerebellar or indirect pathway like spinothalamo-cortical and cortico-pontocerebellar tracts. Sources of information are proprioceptive general somatic sensation from end organs of muscles, tendons, joints, exteroceptive general somatic sensations mainly from end organs for touch and pressure, proprioceptive special somatic sensation from end organs for balance, for example vestibular apparatus, and finally exteroceptive special somatic sensation for end organs for sight, photoreceptors, and end organs for hearing, cochlear apparatus. In stage 2, all sensory informations are analyzed and coordinated or integrated in cerebellum. In stage 3, after integration of all sensory inputs, a regulatory effect is exerted by cerebellum in a subconscious or unconscious state on brainstem, spinal cord, and cerebral cortex for maintenance of balance of body through postural adjustment, maintenance of muscle tone for desired sense of position and movements, and finally coordinated and smooth muscular activities to proper range, extent, and direction. Okay, now let's wrap up this lecture by a very useful video about cerebellum examination.
We are now going to demonstrate the cerebellar exam. The cerebellum can be affected in many of your patients, and you might not recognize that it's affected unless you test the cerebellum. The signs of cerebellar dysfunction from head to foot are scanning speech. So if you ask the patient to say something with many consonants in it, such as the British Constitution, they might say the British Constitution. Scanning speech, breaking it out into individual syllables. They might have nystagmus. So if I ask the patient to follow my finger, they would have a nystagmus with the fast phase pointing to the site of disease. I should also say that if they have disease in the vermis and flocculonodular lobe, the patient wouldn't even be able to sit up. They would have truncal ataxia, and you would not even be able to get this far in the testing. Continuing after doing scanning speech, nystagmus, we would now look for dysmetria with the finger-to-nose test. The whole idea here is to try and put as many joints in play as we can. Just count them, 1, 2, 3, 4, 5, 6, as many joints as we can where agonist and antagonist muscles are working together, and the great beauty of the cerebellum is that it's able to coordinate all of this in one smooth motion. And I'm going to now ask the patient to touch his nose as quickly as he can. Back and forth if you don't mind. Excellent. And now touch my finger in your nose. This task, which is not easy, comes about because of an intact cerebellum. Another test that you can do is called the rebound test. It's called the rebound phenomenon or test of Stewart and Holmes. When I ask the patient to pull on my hand if you would, when I let go, if I were to let go suddenly, the hand doesn't go and hit him in the face because the antagonist muscle quickly stops the movement. But in cerebellar disease that may not happen. So protect the patient by putting a hand across the chest. Pull on my hand, please. One more time. And again, if the patient had cerebellar disease, the hand might go flying up there, and that is why you need to protect them. I'm now going to test for rapid alternating movements. So if you can put one hand out like so, and with your other hand, do this as quickly as you can. Do it on the other side. And this phenomenon, if it's abnormal, is called dysdiadodocokinesia. The next thing we look for is pendular knee jerks. Patients with cerebellar disease might have hypotonia, but it may not be very evident because we're much more tuned to looking for hypertonia than hypotonia. And one manifestation is that when you do the knee reflex, in a patient with cerebellar disease and hypotonia, the leg will keep swinging 3 or 4 or 5 times after you've done the reflex as an indication of their hypotonia. We're now going to step into the hall and do the gait. The cerebellar gait is characterized by a broad stand and by a wide staggering quality to it. People will tend to fall towards the side of their illness, so if the illness is in the cerebellar hemisphere on the left, they might fall in that direction. When asked to stand still, their trunk may sway like this, and that is called titubation. And obviously they would have problems with all the other cerebellar tests. One caveat, many people think of the Romberg test as being a test of cerebellar disease. The Romberg test has nothing to do with the cerebellum. The cerebellar patient is already swaying, and it gets a lot worse when you have them close their eyes. But the Romberg test is really a test of proprioception. When you and I are standing like this with our eyes open, we are getting signals from our joints to tell us where we are in space. If, however, you have a problem with proprioception because of your peripheral nerves or posterior columns, then you're relying on your eyes to tell you where you are in space, and therefore, the moment you ask the patient to stand still and close their eyes, the patient begins to sway. And that is a positive Romberg test. It has very little to do with the cerebellum.